Today's episode begins not with discovery, but with a poem. 1959, Francis Crick, one of the minds behind DNA's double helix, is standing on the California coast reading peyote poem by beat poet Michael McClure. The words full of cosmic unity and vivid hallucination strike something deep in him. Crick, a scientist who rejected religion but never stopped seeking meaning, pins that poem to the wall of his Cambridge home. For him, science was poetry, a way of finding structure and beauty in the chaos of the world. Welcome back to Breaking Math. I'm your host, Autumn Finaf, and today it's the show where we explore the ideas that shape our understanding of our world, from equations to consciousness, and from chaos to code. Our guest today, Matthew Cobb, has written about the side of Crick not just as a scientist, but the seeker. We'll talk about curiosity, creativity, collaboration, and how the search for life's blueprint led Crick to chase the mystery behind the mind itself. Now, what inspired you to write Crick, A Mind in Motion?
Well, I'd written two other books which unintentionally had ended up being all about him. So in 2015, I published a book called Life's Greatest Secrets, which was all about the race to crack the genetic code after the DNA double helix had been discovered, the implications of that, what happened over the next kind of 10, 15 years. And that turned out to be all about Crick, which in fact I should have known it was going to be, but I obviously hadn't done my research properly enough before I started writing. But, you know, this character, his brilliance, his insight just kind of blew me away. And then in 2020, I published a book called The Idea of the Brain. And that was a history of our ideas about what the brain is and how it works and so on. And in 1977, Crick had moved to California. He'd left Cambridge, UK, gone to California, worked at the Salk Institute. And he'd gone to study consciousness. And I knew he'd done this because I was an undergraduate as a student in psychology, in fact, when this happened. And so, you know, everybody laughed, ho, ho, ho. Crick's going to crack consciousness. And spoiler, he didn't. Nobody has yet. But so I knew that he hadn't made any single great breakthrough. But to my great surprise, when I was writing this book about really, in a way, the history of neuroscience, is that in the section, the half of the book that deals with the modern era, he kept on muscling his way into the front of every chapter. And I had no idea that, although he hadn't made any great breakthrough, his influence had been remarkable. And I then started thinking, well, how can you do that? How is it possible? You know, I mean, being really smart in one area, I get that. But being really smart and influential in two areas, how is that possible? How can somebody fit all that in their head? And so I decided I'd write a biography, which I've never written. And I thought that, I mean, there are two biographies that are on the market already, or they've been around for a while, since shortly after he died in 2004. I wanted to write something that was neither an academic biography, which is what one of them is, nor quite a short and snappy version for the general public. I wanted to write something in between. I wanted to really get to grips with the man and his ideas and his life in other respects as well.
Now, you start the story as a poem and not a molecule.
Yeah, that's right.
Why do you think the moment captures the essence of who he was?
Well, yeah, the book begins rather strangely for most readers, which is the point. It starts with Crick in San Francisco in 1958. So this is 5 years after he's co-discovered the structure of DNA. Well, he's a famous among scientists, but he's not known to the general public particularly. And he's working in San Francisco, and he goes to City Light's bookstore, which was the kind of the beating heart of the beat poet movement. And he goes down into the basement, and he finds this long poem printed on a long sheet, and it's called Peyote Poem, and it's by a man called Michael McClure, who at the time was not particularly well known. And this poem describes the effects, well, it's a psychedelic trip, it's the effects of eating the peyote cactus buds that McClure ingested. And it's very vivid, it's very emotional, it's very kind of muscular, it's all in capitals, there are horizontal lines all over it. Crick was really, really taken with this. He'd always been interested in poetry, but this was something way off. This wasn't T.S. Eliot or W.B. Yeats. This was really kind of something else. And I thought he not only bought the poem, he pinned it up in the hall at home. And there was one particular stanza that he was very taken with, which in which McClure is describing the kind of, the effects of this drug on his brain. And he says, "This is the powerful knowledge," in capital letters, italic, underscored. And Crick thought this was marvellous, and he used it as an epigraph later on to a chapter in a book he wrote. And so it was really important to him. And I knew that he went on to become friends with McClure. And I thought this will wrong foot everybody, because everybody expects either you're going to start with a double helix, or you're going to start with winning the Nobel Prize in 1962 or something like that. Whereas I wanted to say something else about Crick, apart from simply talking about his science. And in fact, by understanding what he liked in the poetry, it also gives you an insight into his more intuitive approach to doing science. It wasn't simply a matter of kind of, adding up the numbers in a spreadsheet and saying, oh, that's the answer. He would make intuitive leaps, and this more emotional and subjective side to his thinking I thought was really interesting, and wasn't what I expected to find when I started writing the book, and I don't think most readers would expect the book to begin that way. He did later experiment with LSD and maybe with mescaline, which is the active ingredient of peyote. It's not quite clear, but he clearly, at the time, he didn't know what peyote was. He'd have known about mescaline because he would have read Aldous Huxley's The Doors of Perception, which came out in 1952. But he wasn't, this wasn't something that he recognized. I think that's why he found it so exciting, that it was completely kind of orthogonal to his everyday experience, or in the mysticism, the kind of Celtic mysticism of W. Yeats, the Anglican mysticism of T.S. Eliot, or the Catholic mysticism of Gerard Manley Hopkins, all of whom were poets whom he greatly admired. But he found their religious beliefs rather frustrating, whereas this was something completely different. This was something that was rooted, and because McClure was, like Crick, a materialist, it was rooted in biology and science, but it was also quite weird and quite exciting. So later on, when he did indulge in various mind-bending drugs, he kind of recognized the accuracy of what McClure had written about.
Now, Crick once called himself averagely bright. Do you think that humility was sincere, or part of how he framed intelligence itself? This can start at an early age for self-perception.
Yeah, I think that's a very interesting point. I mean, I think it was true. I mean, one of the things, biographies you start on the born. He's born in Northampton on 8th of June 1916, and then you try and see what's going to happen in the future. I don't know, with somebody like Mozart, by the time he was 5, he was entertaining the emperor and composing symphonies and stuff like that. So clearly he was marked out from the beginning. But Crick wasn't like that. He was. I think, it's true. He said he was averagely bright. He did okay at school. He was known for being kind of curious and asking lots of questions, but that's what children do. So I don't think he was being modest. I think he was being quite accurate that he was smartish, but there was nothing special about him. And indeed, right up until so he goes to university, does a degree in physics and maths to University College London, where he gets not the top mark, which will be a first-class degree. He gets what's called a 2-1, which is still good, but it's not somebody who's blown everybody away and has, aced all his exams and all the rest of it. It's somebody who's smart, but nothing special to write home about. He starts a PhD on what he described as the most boring subject in the world, which is how water changes its viscosity under pressure. And then the war breaks out, and he gets conscripted into the Army, or the Navy Scientific Service. And that's when he begins to change. That's when you can see something happening to him, and he starts to think in a rather different way. So I think as a child, he was averagely bright. As a man, I think he was unbelievably smart in some respects. Not in every one. As in the 1960s, somebody chalked on a wall in Cambridge, England, "Crick for God," and he wasn't God. I mean, he was very very clever, but there were distinct limits, and we'll maybe talk about these later on, to his understanding of things and his grasp of things, which in fact was quite nice to discover. So there were moments when I could think, hey, Francis, you really don't get this. And then you ask the question, why is that? How can you be so smart in some areas and so just really don't get it in others?
I think the term is coined genius zones. Everyone has their own genius zone, but
Okay, that's nice.
Yeah. So if you teach a fish to walk outside of water, you're not going to have the same result as telling them to swim. So everyone has their own exact intricacies for brilliance, right? But you also mentioned that he worked in the Navy, and if I remember correctly, it was naval mines during World War II.
Yeah.
How did that shape his collaborative instincts later on?
Well, it wasn't so much the work he was doing, which was kind of intricate. It was designing circuits to enable. I mean, they were kind of in an arms race, literally and metaphorically, with the German Navy. So the point about a mine is that you want it to be able to explode if it comes into contact or close to an enemy ship, but you also want to be able to defuse it if it's one of your ships that it's close to. And so you had to develop circuits that would enable this to happen. And every time the Germans captured one of these mines, they would change their approach to enable them to be able to disarm it. So there was this very rapid turnaround. You know, they discovered that their old techniques didn't work, and so they then had to develop new circuits. The key point was not so much the work he was doing, but he met a mathematician, a very odd mathematician who went on to play a really important in his part in Crick's life, a man called George Kreisel, who was not only a mathematician, he was a logician, got his degree from Cambridge in 2 years. Normally it would be 3. So he was very, very smart. And he worked with Wittgenstein, the philosopher and philosopher of logic, and argued with him. And Crick learnt from Kreisel because he's a kind of a mixture between a mathematician and a philosopher, which is infuriating, right? Because these are people who will ask really, really precise questions and make you think about what you're thinking about. They won't just take any old flim-flam. They may not know what you're talking about, but they've got ways of making you think more clearly. And he always argued that was what changed his mind, literally. That he said something like, before I met Kreisel, I was a very woolly thinker and, full of kind of jokey aphorisms, a bit like kind of Oscar Wilde kind of thing. And now he met this man with this absolute insistence on rigor, logic, precision. And that helped Crick do 2 things. Firstly, change his way of thinking. Secondly, as you suggest, their interactions became model for what characterized Crick for the rest of his life. That is, he never worked on his own. He was always working with somebody else with whom he could argue, debate, struggle to try and find the answer. And that became his way of working right up until the day he died.
Now, he was always collaborating with someone. When he was becoming a biologist at Strangeways, Crick was surrounded by living cells, and he quickly learned that they didn't obey his rules in physics.
Yeah.
What did that experience teach him about complexity, and who was he working with at the time?
Yeah, so Crick was after the end of the war, he worked briefly in naval intelligence. We don't know what he did there, but there are various documents in his archives stamped top secret in which he was kind of making politics about trying to reorganize the intelligence services. But in the end, he decided that wasn't what he wanted to do, and he wanted to go back to science. And in 1947, he wrote a little document which is really quite remarkable, in which he sets out what he wants to do in life. And he says, I want to solve 2 things. I want to understand what life is, and I want to understand how the brain works, what consciousness is. And both these things have had in the past, and to an extent still do, mystical explanations. People will reach for some kind of a mystical or divine explanation for what's going on. And Crick wanted to root both these mysterious and astonishing things in material science, in material reality. Amazingly, he did in fact go on to do both those things. So the first thing he decided he wanted to do was he needed to get training, as you said, in biology, because he trained in physics, didn't know anything about biology. And this really is biology. And he gets a grant basically just a training grant from the British Medical Research Council. And he goes and works at a place called the Strangeways, which is an institution just outside Cambridge. And he's working on motility in cells. They're trying to understand how cells move and their internal structure. And it's not very exciting, but it's very hard. He said later on that, it required a lot of green fingers because he was growing these single cells in cell culture. And as anybody who's ever done any biology knows, it's really difficult to keep the things you're studying alive. Whether they're ants or flies or plants or single cells, they need care and attention and so on. And he probably wasn't very good at that. Indeed, he generally wasn't a very good experimentalist. That was not his forte. So this is 1947, and then he learns that through his friend Kreisel, who's a bit like Zelig, that film in which this character keeps on popping up. Kreisel is like this. At all these key points in his life, he actually knows people or he introduces Crick to people or whatever. But Kreisel mentioned Crick to a man called Max Perutz, who was an Austrian refugee who had set up a Medical Research Council unit into biophysics, which is using the techniques of physics to study biology. And this was a big thing after the war, partly because lots of physicists had fallen out of love with physics because of the bomb. So this science that they had loved had now produced this terrible destruction. And so there was a big move out of physics into biology, with these very clever physicists wanting to use these techniques to understand how life worked. And Crick decided, okay, that's really what I want to do. This business with the cell culture, that's not really my thing. I want to go to the MRC Biophysics Unit, which was in the Department of Physics in Cambridge, in the Cavendish Laboratory, and use what's called X-ray crystallography, which is a technique of bombarding crystals with X-rays, and you get these amazingly complicated patterns. Then you've got to do some really complicated maths and thinking and a bit of chemistry to try and work out what the underlying structure is. So this is basically his second research career. At this stage, he's 33, 34, and he's already abandoned one PhD. And now in his mid-30s, he's starting another one and a new career. And really, that's kind of the ground zero for the Crick we know now, because he's now using these techniques. He's got this method, this very intense way of arguing and discussing, which he's acquired from Kreisel, and he's in Cambridge at this laboratory run by Max Perutz.
Now, at some point, he discovered Schrodinger's What is Life? What did that unlock for him that physics alone could?
So, Schrodinger was a Nobel Prize-winning physicist who had fled the Nazis in Austria when they invaded Austria in 1938, and had ended up in Ireland at the Institute of Advanced Studies in Dublin. And part of his job was to give a lecture to the public, because he's got this prestigious, this fancy post. And being a physicist, he thought, okay, let's do something easy. I'll tell them what life is, right? Because that's obvious, I mean, I'm a physicist. I can work this out. And so he read lots of books and articles from the ideas from the 1930s about how life worked, about the nature of genes and so on. And he gave what was supposed to be one lecture, but turned into three. And this little book of lectures was published in 1944. And it was devoured by many, many people, including Francis Crick, Jim Watson, Maurice Wilkins, who we'll talk about in a little while, who's ended up working on DNA, who had been on the Manhattan Project and was one of those physicists who had really, really fallen out of love with physics because of what he'd been working on. And now here we've got Schrodinger saying, look, we can use the techniques of physics to try and understand, say, the nature of the gene. And this was immensely appealing, obviously, to the physicists, because they thought, okay, we can use this stuff we understand to understand this really, really complicated thing. And what Schrodinger held out was the idea that maybe it wasn't going to be so complicated after all, and you could resolve these issues. So Crick found that intensely exciting. The other thing that really excited him was a little article he read by Linus Pauling, who was a chemist, who talked about X-ray crystallography, which was really a big thing in the UK at the time, and with the work of J.D. Bonal in particular, and Dorothy Hodgkin, who would go on to win the Nobel Prize in 1964. And Crick decided, these two sources of information, of ideas, Schrodinger's book and a little article by Linus Pauling, they convinced him that this is where he wanted to go. And that was the kind of switch, because these tools, you thought, would be able to understand, say, molecular structure, the molecular structure of biological molecules, and perhaps that would give you eventually an understanding into how a cell worked.
Now, then he met somebody that would change his life, right? Jim Watson. brash, young, impatient, and he's exactly the opposite of Crick. Together, they would decode life itself. Now, what was this dynamic between Watson and Crick like in those early days?
Yeah, so, I mean, you got to remember that none of this happened the way we imagine it did. Crick is there in Cambridge studying the structure of proteins. That was his PhD. He's got to finish his PhD. He's only got a certain number of years to do it. He's got to finish it in 1953. And really what he's supposed to do is studying the structure of proteins using X-ray crystallography. Jim Watson was, as you say, this absolutely astonishing young man. He went to Chicago University age 15, and he had his PhD by the age of 21. So incredibly bright, very ambitious, a bit odd as well. And he, in various convoluted ways, decided that he wanted to use X-ray crystallography to try and understand the structure of the gene. But the route he was going to do that was by studying viruses, because viruses could be thought of as basically just a gene, and he wanted to understand the structure of viruses. So he was able to wangle his way to the Cavendish in autumn of 1951, and Watson and Crick hit it off virtually straight away, partly because they're both very different in some respects, their personalities. But in terms of their approach and their fascination and their conviction that, in fact, quite complicated problems could be solved using these techniques, that's what united them. Now, it must be said, Watson knew nothing of this. He knew nothing about X-ray crystallography. Yeah, I mean, he trained he was going to be a bird watcher. He had a zoology degree. And, Crick described him to his friends when he explained their work after the discovery of the double helix, that he's a zoologist. So he's not somebody who's like Crick is trained in physics, or Wilkins had the same background. He's somebody who's come from a very different angle. And so this was very hard for him because it's very mathsy, it's very physicsy, it's a difficult technique. But they liked chatting about things. And Crick, like nothing else, as his boss put it, he likes finishing other people's crosswords. So if somebody's got a problem, if you talk to Francis about it, he'll solve it. He'll work it out, because he's got this kind of mind. He likes kind of, a bit like a magpie or a jackdaw. He's going to always looking for something new and shiny and find something out. And that led to the whole hoo-ha about DNA, and led to the many misunderstandings that still circulate on the internet today about what exactly happened. But I think the thing we need to understand is neither of them was supposed to be studying the structure of DNA. That was not their job. It wasn't what Crick was studying, and it wasn't. Watson just didn't have the tools to be able to do it, even if he'd wanted to, but he was supposed to be working on his viruses. So this was not their job. This was not their problem.
So at some point, apparently they stole data, Watson and Crick. The story of Rosalind Franklin's Photo 51 has become a flashpoint discussion in the ethics of science. How do you approach this controversy today?
Well, I approach it like a historian and a scientist. So what actually happened, and how do we know? So, yeah, everybody knows, they don't think, they know that Watson and Crick stole Rosalind Franklin's data, and that gave them the double helix. There's an internet joke. What did Watson and Crick discover in 1953? Rosalind Franklin's data. So it's quite a good joke. But you need to ask yourself, well, how do I know this? Where's this story come from? And secondly, if Watson and Crick stole her data, how were they able to see something where, Crick was very clever, Watson knew nothing about this technique? How could they see something that this very experienced X-ray crystallographer, Rosalind Franklin, could not? So the story, in fact, is a bit odd. And when you actually dig into it, it kind of all evaporates and disintegrates in your hands. The only reason people talk about photograph 51, this very famous photograph showing the X of the structure of DNA in an X-ray crystallographic image, is that Franklin published that. Watson and Crick published the structure of DNA. There were 3 papers published together in Nature magazine, one after the other, and they were bundled together as a whole bound set because they were all complementary. One was from Watson and Crick, which just described the structure with no evidence whatsoever. Then there was an article by Maurice Wilkins, who was the head of another MRC biophysics unit, which was the deputy head, another MRC biophysics unit, which was in King's College London. And the third article was from his colleague, Rosalind Franklin, who was a postdoctoral researcher who'd come with a grant, and the people at King's had suggested to her she might want to study DNA. It wasn't her idea at all. And unfortunately, for all sorts of reasons, Wilkins and Franklin did not get on. They were completely and utterly different, not in the way that Watson and Crick were, but in a bad way. So Franklin was very vivacious. She loved to argue. She came from an intellectual Jewish family where you'd argue about all sorts of things. She spent her whole time in 2 or 3 years in Paris where she'd learned X-ray crystallography. She loved that because the French will argue about anything. And so this was her very outgoing, vivacious way of discussing things, of playing devil's advocate if somebody had a position to try and find the weakness in it. So, a bit Crick-esque in some ways. Whereas poor old Maurice Wilkins was really shy, retiring. He hated debate. He hated argument, confrontation. He'd turn away from you when you were talking. So these two people were like chalk and cheese. Furthermore, Wilkins thought that Franklin was there to help him. Franklin had been told the problem of DNA structure would be hers alone. So this was the head of the lab, had done something very, very bad and caused confusion, and it was never sorted out. It was, so they couldn't resolve. If they'd been able to work together, so that shows you why science is about people. If Franklin and Wilkins could have worked together, then we wouldn't be talking about Watson and Crick. I wouldn't have written this book because Crick would be an unknown person or a very minor figure. Unfortunately, they couldn't, so the problem was split up. It's like keeping, warring children apart. They were separated. They don't have anything to do with each other. Part of the problem was that the man I mentioned earlier, Linus Pauling, he got interested in DNA, and he was a really, really smart bloke. He's an American chemist, and he, in the winter of 1952, by which time Franklin has decided, I hate this place, I hate Wilkins, I can't stand any of them, I'm moving, I'm going to taking my money, because it was her money to go and work in another laboratory in London. She was going to have to leave the DNA problem behind because it wasn't hers, it was the lab's. In end of 1952, Linus Pauling submits a manuscript, or writes a manuscript about the structure of DNA, which is completely wrong. Watson and Crick know it's wrong because it was a triple helix, kind of inside out, virtually identical to one that they'd come up with when they'd briefly toyed with the idea 2 years earlier, in which Franklin had taken one look at and laughed at and said, that's just wrong. It's just complete nonsense. So this meant that although Watson and Crick had been told not to work on DNA by Sir Lawrence Bragg, the head of the MRC unit in Cambridge, because he was fed up with these people causing him problems, because their initial attempt in December 1951 caused a huge row with the people in London to say, what are you doing? This is our molecule. Why are you fiddling around with it? Plus you're wrong, and you're stupidly wrong. And
scientists are so dramatic.
Well,
people do not realize this.
That's right. I mean, it was, it's very important. So it was an inter-laboratory, inter-university row. And, Bragg didn't care. He couldn't stand Crick because he hated his voice and he was, like I say, always finishing somebody else's crosswords. He annoyed Bragg intensely. Watson he barely knew of his existence. And these two nobodies were causing this diplomatic row. So basically he said, look, get on and do what you're supposed to be doing, which is more or less what happened throughout 1952. And then when they hear that Pauling has made this really bad mistake, bizarrely, this leads Bragg to say, OK, you can do that. And this is because Pauling had scooped Bragg and the Cambridge people over a really boring structural thing. It doesn't matter what it is. About a year earlier, Bragg had a beef with Pauling. And now, if Pauling had this is Watson convinces him. Watson goes and says, look, Pauling's made this mistake. He's going to realise it's a mistake. We've got a window, a short period of time in which we can crack it. Me and Crick, just leave us alone for a month or so. We'll crack it. And basically, that's what happens. And the story of Photograph 51, we're going to get there. There you go. Watson goes down mid-January to London, to King's, to show Wilkins and Franklin this manuscript to say, hey, look, Pauling's on our tracks, or he's on your tracks more to the point. And at some point, Watson sees an image of DNA, a very clear image of the DNA molecule. Now, people say this is photograph 51, but if you actually read Watson's account, he doesn't say that at all. He says Morris shows me some images, and Morris, he says, have been repeating some of Franklin's experiments. So although Franklin did make this very good image, she literally, after studying it briefly, she put it in the drawer because it wasn't the issue that she was interested in. All this is explained in the book. It's too complicated to go in now. So she actually looked at it and thought, yeah, okay, so DNA is some kind of helical structure. That's not the kind of DNA I'm interested in. I'm interested in the crystalline form, which is much more complicated. Puts it away, doesn't study it. Now, people have put 2 and 2 together and made 17 million here and said, aha, Watson stole this photograph 51 from Franklin. There's no evidence that he actually saw it. Furthermore, Crick never saw it. And the information that it contains was no different from a little sketch that Wilkins had sent to Crick about 8 months earlier in a letter in which he sketched out a result that he'd had. His photo wasn't quite as pretty, but it was basically contained the same information. So none of the story makes sense. What is true is that Watson and Crick asked Wilkins, they said, is it okay if we start trying to solve this? And Wilkins was a bit of a dope, sort of said, a bit like Eeyore from Winnie the Pooh. He said, oh, well, all right then, okay, if you really want to. He didn't say, look, I need to go and talk to Bragg, the head of the lab, or we need to talk to Franklin. He just said, oh, all right. And what they didn't do was to ask Franklin, is it okay? Now, Franklin was kind of finishing up the project. She was let's say, moving on to a different project, different laboratory. And there's this little note that myself and Nathaniel Comfort, who's writing a biography of Watson, that we discovered, in which she, via a friend of Crick's, was a PhD student called Pauline Cowan, she invited Crick to come to a talk she was going to give and says, but on the other hand, it's not going to be at a very high level, so you can always ask Max Perutz if you want to know more. And Max Perutz, a month earlier, had been at King's and as part of an official MRC site visit to see how things were going. They all went and saw each other's work to improve communication. And he had a report which contained a little paragraph summary, or a couple of paragraphs, from each of the main researchers. And there was data in there from Franklin. Now, Crick and Watson were given this report. Apparently, the implication is that Franklin either expected it or didn't particularly care. However, even that wasn't used. When Watson describes it very briefly in the Double Helix, which if listeners haven't heard, they really should read. If they haven't read it, don't believe everything that's in it, but it really is a cracking read. But in the 1960s, Crick gave a load of interviews to his first biographer, a man called Olby, in which he describes their process. And if you read very carefully their articles describing the structure, it's clear as well. Basically, they spent a month fiddling about, right? They just tried to see, okay, we know what the component parts are. How do they fit together? And this wasn't with a big kind of three-dimensional model. It was all with little bits of cardboard. So they work in 2 dimensions, had these 2-dimension bits trying to work out how many times the spiral might go round, or how they could fit together. And eventually, well, they were getting it all wrong. Nothing worked. A guy in the office said, oh, by the way, those structures that you're using, they're wrong. And they went, no, we got this from the textbook. And they said, oh, well, he said, well, that's wrong. And ironically, Rosalind Franklin was also using the wrong forms. So they then had the right forms, and they wouldn't fit together, the kind of the rungs in the ladder of DNA. And Watson describes that it's on Saturday, the 28th of February, he's got these 2 forms like this. And then one of the bits of cardboard, he turns it over. And now he sees that the bases, as they're called, can fit together. 2 rungs fit. And the 2 groups of bases, A and T and C and G, bind together and they form identical size lumps, molecules that can then fit between the rungs. So basically that's the answer. They got the answer. And then Crick says, look, it's got a particular, what's called symmetry, particular way of organization, which is something that Franklin had recorded and noted, but hadn't thought through the implications of. She said later she could have kicked herself for not thinking of it. So this is used as, Franklin's data is used as confirmation of the model that they've got. But the model, most molecular structures don't tell you anything unless you really, really understand. It's just bizarre, lumpy stuff. This is different. You can explain it to a 6-year-old or a 10-year-old at least. And it's immediately obvious how it copies itself and how the order of the bases can be of any order. And so you've got effectively the space, literally and metaphorically, for a code in there, genetic code, as Crick called it.
After the double helix came into frame, The discovery was never an end point, it was just the beginning. So you describe Crick's mad sessions with Sidney Brenner as loud, hilarious, and wildly productive. What made this collaboration so special?
Yeah, so Sidney Brenner joins the MRC laboratory in Cambridge in 1956. And he's a young South African trained as a physician, but became absolutely convinced that he wanted to be a molecular biologist when, as a postdoc at Oxford, he came to Cambridge to kind of see the structure. It was a bit of a pilgrimage. Everybody in spring 1953. And he takes one look at it. He says, my God, I want to work on this. And he eventually meets Crick in the USA at a meeting, and they really hit it off. And he then comes to Cambridge in 1956. And for the next 20 years, they shared an office. They shared a blackboard or several blackboards, which was really important. And every morning for a couple of hours, they would just yak away. They would talk to and at each other about all sorts of stuff, anything that they'd read or ideas, crazy ideas, and each would elaborate an idea and develop its implications, and then the other one would try and shoot it down. So it's this kind of jousting intellectual that went on perpetually every day that they were in the lab together. Brenner was a much better experimentalist than Crick was, but even so, they, for example, mRNA. So everybody knows about mRNA. It's this kind of intermediary between the DNA sequence and the protein sequence. It turns the genetic message into a protein, or it helps the cell to do that. Brenner and Crick suddenly realized there was all sorts of bits of information about strange forms of RNA that people were finding in cells. And on one very famous day in April 1960, when in fact some other people were present, a group got together in a kind of after meeting after a conference. They were in Cambridge and they're just chatting away, and then suddenly the pair of them go off on one and realize that all this information, these various disparate studies that have been floating around and nobody's really put together, they can now see that if messenger RNA is this messenger, because it wasn't called that then, if it was this intermediary, that it would then take the DNA message and, like a tape on a tape recorder, they say, then put it through a structure called the ribosome, and that was where proteins would be assembled. So Crick in 1953 had come up with the idea of a genetic code, and he used it much more explicitly, or he talked about genetic information. That's ultimately what the whole of life was. It was all about information, whether it was embodied in a DNA molecule or in a protein. Life was information. Now he could actually see a part of this information flow, that this molecule was now considered as being the carrier of information. This is incredibly modern. And Crick and Brenner basically kind of yak at each other for about 10 minutes. And a French researcher called Francois Jacob describes this in his memoir. I quote it in the book about the pair of them, yabbering away. And poor old Jacob, his English isn't that good. He can barely understand it. And that evening, once they've kind of come down from this high of realizing what's going on, Brenner and Jacob decide they're going to go to America, which had the right kind of equipment to be able to test this hypothesis. And they start to work out experiments in there. This is at one of the Crick's famous parties. I don't think it was quite as louche as some of their parties, but anyway, it was a good party. And they're in one room scribbling around, and Crick keeps on coming in and saying, oh, you want to do this, or what about that? So that was the kind of thing that he and Brenner were able to do. They had a number of experiments that the theories and fundamental understanding about how cells work that came out of those mad sessions, as Brenner called them.
He also goes into consciousness. What drew Crick to the problem of consciousness, and did he see it as a continuation of the same logic that he once explained in DNA?
So in 1947, when he comes up with these two things he wants to study. Life is one thing, so he hasn't really solved it, but he's gone a long way. And secondly was consciousness. And what bugged him about both these things was the fact that they were shrouded in mystical ideas. So that, in an attempt to root in materialist science these very complicated biological phenomena, that was what was underlying them. Now, his turn to consciousness did not succeed. I don't think there's any way of putting it any better than that. We haven't cracked the problem. And indeed, it's getting worse. There are now 250 theories of consciousness, which shows that, in fact, the field has lost its way. they're not even talking about the same thing. It's just that it's a big, big mess. But what Crick did was to focus people's mind by saying, if we're going to try and understand this, because he was doing this, as I say, when I was a student, and I remember us sitting around in the coffee lounge going, well, consciousness, it's some kind of epiphenomenon, it's emerging from neurons in the brain, we're never going to understand it, who cares, let's do something else, right? Which, now Crick was a bit more incisive than that. He thought, that's true, but we can ask questions. We can say, which are the neurons that are associated with consciousness? Because not all the 80 billion neurons in your brain are associated with conscious activity. Which are the neurons? Where are they? What do they do? To try and understand what might be specific about them compared to other neurons which aren't involved in consciousness but are involved in other aspects of processing. So that framework, that materialist framework for how to study consciousness, which he began to develop over the next 20 years until his death, with a man called Christoph Koch, who, like Brenner, was much younger than him. Virtually all of Crick's partners, they were other. They weren't like him, right? They were generally younger than him. They were often not from the same country. They weren't necessarily English. Most of them were men, the ones he had the most intense relationship, but he did have very close relationships and intense relationships with a number of female scientists, in particular in his neuroscience period, but also with Rosalind Franklin in the second half of in the late 1950s. And what Crick does is to try and this is why he kept on popping up in my book on the brain. He says, one, we can have a materialist approach to consciousness, but we need to understand much more about structure, about how the brain is organized, not in broad tracts, I've got this big lump here and this big lump here, but how the cells are organized. And this was quite unheard of at the time. But basically, he's at the origin of the whole kind of current movement for connectomes. That is trying to understand, organize at a cellular level, describe at a cellular level, the connectome of a brain. We haven't got anywhere near the human brain. The best we've got is the connectome of a fly brain, which is about 10,000 neurons. Okay, so the human brain is 80 billion, the mouse is about 70 billion. So we're way, way off, right? But this is what Crick said we needed. Absolutely right. He also made various predictions about the kind of techniques we'd be able to develop. He predicted, for example, that we'd be able to control, if by manipulating cells genetically, we'd be able to control neurons with light. We'd be able to use light to control their activity. And indeed, that's exactly what happened. I mean, he published an article on this, and within about 6 months, somebody sent him an email saying, hey, that's exactly what we've just done. So he's thinking about how techniques can help us move forward. But above all, it's this materialist framework for studying consciousness, which he expressed not only in lots and lots of articles in leading journals, in particular Nature, but also in a very successful popular book called The Astonishing Hypothesis, which he published in 1994, and which really helped ignite the public's interest in this question. So, there are books on consciousness everywhere now, and it's all Crick's fault.
As you were going through some of his research or finding out more information about him, I know you already have a couple of books on Crick. Was there anything really interesting or surprising that you found this time around?
Well, I think firstly, the collaborative aspect became much clearer to me, and that was the starting point of the book. In fact, I wanted to think about how he and Brenner were able to have this incredibly productive relationship for the 20 years. And then, as I was working on the book, I realized that in fact this was an approach that he used consistently. And I thought that was very interesting because although scientists tend to work in teams, having that very intense, almost intimate relationship with somebody who you trust, because, you're going to expose yourself to ridicule by saying something stupid, right? And they're going to shoot you down. So you've got to trust them. You've got to like them. You don't necessarily need to be best mates, but you've got to be very open to their ideas and their way of thinking and trust that they're not going to, completely take the mickey. I thought that was very interesting, and how, from a kind of science policy point of view, could you cultivate this? Is this something you could teach, train people to do? So that was kind of the starting point. And then discovering that it was so systematic in his way of thinking, I thought was very interesting. And then the other thing, which is connected, is where we started with his interest in poetry and the intuitive side of his thinking. Because although he was logical and a scientist and all the rest of it, he was also incredibly intuitive, and he would make leaps or be adventurous, as he put it. And this meant that a lot of his, he'd throw out ideas. He published articles, he published a theory of dreams in Nature, which was wrong, but was very, not of the content of dreams, why you dream of your Auntie Jessie as a giraffe or something, but why we dream at all, what's going on. And this idea he developed in parallel to the developing ideas of computational models of behavior, which in fact led directly to today's AI models and so on. So he's really coming up with ideas partly to enable them to be shot down. He expected most of his ideas to be wrong. These weren't theories of everything that explained everything. They would be effectively a hypothesis, one particular thing. Okay, how could we test that? Experimenters, here's my idea. Can you see if it's right or wrong? You go away and see whether this particular part of the brain is involved in it, in a particular behavior, or whether the particular neuronal mechanism that he predicted for dreams, does that actually occur during dream sleep, and so on. So I thought that was intuitive aspect was very interesting, and it links with what he liked in or admired, what he felt in reading McClure's poetry, this very muscular, it's a kind of materialist mysticism. You can go and watch him reading on the internet. He went to San Diego where the Cricks lived, and he gave a reading in 2000. It's on the internet. You can watch it on YouTube. And you can just see Crick at the beginning and the end, shaking his hand and so on. But McClure's kind of muscular, mystical, psychedelic experience, that struck a chord in Crick's way of viewing the world and trying to understand, the perception. When you've got a set of data, you know there's something in there. You intuit it, you can feel, but you can't necessarily make sense of it. And you kind of try to explore it. How could I be convinced that this interpretation is correct? What would I need to do next? And this very subjective approach, which he also found in McClure's poetry, I thought was very interesting. And it made him not a very smart robot. He's not Commander Data, right? He's somebody, he's a human being with these all sorts of passions. He had very interesting personal life. He and his wife had no open relationship, which undoubtedly didn't always go well, but was quite modern in some respects. Young people might find it quite interesting that this person back in the late 40s was living a life that they might think was the best way of having a coupled relationship.
Last question. Is there anything that you want someone to take away from this book? What's the biggest thing?
So Crick also got things very wrong. So he was an advocate of a kind of class-based eugenics, that he's thought that the state should encourage rich people to have more children and poor people to have fewer children. And you should perhaps tax children. Maybe that's one way of doing it. It's a very clever idea. So he has these very kind of childish Edwardian ideas from these ideas that he probably learnt in the 90s as a young boy, which were very fashionable at the time, and never really thought about it. He wasn't interested in politics. He didn't read the newspaper. He didn't have a TV or a radio until the 1980s. So he's completely disconnected from most things that many people would think were important. And this is, he clearly just does not get it, which is quite fun because it means I can read this stuff and say, Francis, you're an idiot. Why are you saying this? This is just not true. Why do you believe this? Why aren't you questioning your beliefs in the same way as you would do about other issues, about scientific issues? So I think what I want the reader to come away with is that he's this complex character. He's got these sides of his personality which are mistaken and pretty unattractive today. But these aren't overwhelming, I don't think. His overwhelming contribution to science, both as a molecular biologist and as a scientist. And that's really what the book is about, trying to get inside his head. And also, therefore, how these old-fashioned ideas can co-inhabit, these old-fashioned, kind of socio-political ideas, can co-inhabit in his head with basically the most incredible modern ideas. And it struck me that, that feels odd. But it only feels odd because he's so modern that you assume he must be modern in every respect. He was born in 1916, so he's as old as my father. many readers who'll be as old as their grandparents or great-grandparents. And, I don't suppose great-grandparents would agree with their great-grandchildren on lots of things, So he's in a way, he's a man out of his time. He's stuck in some respects in the 1920s, in the early decades of the 20th century. But his ideas about science are influencing today and tomorrow, for the rest of this century at least, will carry on shaping how we think about the natural world.
Matthew, thank you so much for coming on Breaking Math. Now, Francis Crick's story is often told as a tale of one discovery, DNA. But in truth, it's a story about never stopping at the first answer, about finding beauty in structure, meaning in molecules, and poetry and patterns of the mind. Now, he once said that science was the only permanent revolution, and maybe that's what his life really proved, that curiosity itself is immortal. So as always, stay curious and stay informed. Until next time on Breaking Math.